Hello viewers, welcome to this new session. My name is Kamran and in this video I will talk about the electrochemical biosensor. Before going further, I would like to introduce biosensor first. So biosensor is an analytical device which is used to determine the presence and concentration of a specific substance in a biological analyte, and biosensor made of a biorecognition element which is also known as bioreceptor and a transducer. And in simple terms, it can be defined as it is device that interact to specific bioanalyte with the help of biorecognition element, and this interaction produce physical or biological change which is detected and converted from biological signal into electrical signal with the help of transducer, and this signal then processed and is shown up on a display screen. And the main purpose of biosensor is to provide rapid, real-time, accurate and reliable information about the analyte. So this figure represents the biosensing process, where red dot represents the analyte and green one represents the biorecognition element and the white strip represents the transducer. Biosensor can be classified in two ways. It can be classified based on biorecognition element, and it can be also classified on based on transducer. Biosensor based on transducer further can classified in three ways. That is optical biosensor, electrochemical biosensor, mass-based biosensors. But in this video we will talk about the electrochemical biosensor. Electrochemical biosensor are the biosensor that transform the biochemical information such as analyte concentration into an analytically useful signal such as voltage, current, resistance. From figure, it is visible that when analyte comes in contact with the biorecognition element such as enzyme, an electrochemical reaction takes place. And after that, a transfer of electron occur. That means the ionic concentration of analyte and the pH values also change. To measure the target analyte in electrochemical biosensor, three types of electrode are used. Those three electrodes are the working electrode, which is also known as sensing electrode, and second is counter electrode, and third is reference electrode. Among these three, a working electrode is very much important because it is immobilized with the biorecognition elements such as enzyme, urease, etc. Electrochemical biosensors are further classified in two ways. First is the amperometric biosensor, which measure the current, and second is conductometric biosensor, which measure the conductance of the sample, and conductance is the reciprocal of the resistance. Amperometric biosensor are the biosensor which measure the current arises during the electrochemical reaction. The basic component of amperometric biosensor are three electrodes, namely working, counter, and reference electrode, and the monitor. Monitor is used to measure the current. The working electrode is immobilized with the enzyme such as glucose oxidase, and the and when analyte comes in contact with the working electrode, and oxygen diffuses to the working electrode, and oxidation reaction results in the flow of electrons from the working electrode to counter electrode. This flow of electrons constitutes an electric current which is proportional to the amount of target analyte. Enzymes typically used in amperometric biosensor are oxidase that catalyze the following class of reaction. The typical example of the amperometric biosensor are the glucometer, which is used to measure the glucose present in the blood. So it is basically contained with the two part. First is the test strip and second is monitor that measure the current. So here is a figure of the test strip. It can be seen that. So here is the detailed view of the test strip, where it can be seen that there are three electrode. First is working, counter, and reference electrode. To know detail about the a glucometer, I will like to introduce the basic principle of glucometer. So glucometer has two major components. First is test strip that contains glucose oxidase enzyme, and the blood is placed on the strip to react with the glucose and create a current. And the second component of glucometer is the monitor that contains electrochemical device that measure the current arises by reaction between the blood glucose and glucose oxidase enzyme in the test strip. Based on the working principle, the more blood glucose present in sample, the stronger voltage generated. The time taken for reading to generate the current is approximately 5 seconds for electrochemical method, making this blood glucose sensor device is more efficient. This is the reaction that takes place on the street. So here it can be the beta D glucose which is present in the blood is react with the glucose oxidase in presence of oxygen, and it after the reaction it forms a D-glucono 1,5 lactone and the hydrogen peroxide, and it is further hydrolyzed, and finally glucono acid is formed. This reaction generates an electrical current that forces electron to flow between the working electrode and counter electrode. With this method, an impregnated enzyme is present so that when contact with the glucose, a current is generated. Conductometric biosensor. Conductometric biosensor are the biosensor which measure the conductance of the sample or medium. Conductance is directly related to the amount of ion, which means target analyte in the solution or sample. And since many enzymes reactions result in change in ion concentration and pH, they are very much suitable for the conductometric biosensor. And the main components of the conductometric biosensor are two electrodes, namely working and counter electrode, and the monitor. And working electrodes are immobilized with enzyme, namely called urease. So when a sample comes in contact with the working electrode, an electrochemical reaction takes place, and the transfer of electron between the working electrodes and the counter electrodes is occur, and the pH of that sample is also changed. It means the resistance of between the both electrodes is changed. Since resistance is the reciprocal of conductance, which is further measured by the monitor. So here conductance change is represent the concentration of target analyte in sample. So the unique characteristic of the enzyme catalyzed reaction studied by conductometric method is the change in the solution resistance. It means reciprocal of conductance during the electrochemical process. The best example for the conductometric biosensor is the urea detector, where biorecognition element is urease, which is immobilized on the working electrode. Here is the uh, typical representation of urea detector. So it can be seen that there are two pairs of the electrode. One is represent the working electrode, another one is counter electrode. So when solution or sample comes in contact with the working electrode, an electrochemical reaction takes place, and transfer of electron is occur, and pH of between the pH of that solution is also changed, and resistance between the both electrode is changed. It means the conductance is changed also, since conductance and the resistance are reciprocal to each other. So here by using monitor, that change can be measured. So this here is a chemical reaction that uh, take place during the detection of urea. So it can be seen that the urea is react with the water in presence of urease, and it forms NH4 plus and CO3 and OH.